Welcome to Trial Site News Podcast Series. Today we have Sonny Hsiao, who's the founder and CEO of Acepodia. Founded in 2016, the company seeks to develop new therapies in immune-oncology and cell therapy medicine. Put another way, Acepodia is a novel technology platform that serves to arm immune cells with antibodies to cure cancer. So, Doctor, welcome.
Hello, everybody. Happy to be here.
Now, for our Trial Site News audience, could you expand on your company's strategy to use novel technology platform to arm immune cells with antibodies to cure cancer? What does this mean, and what are the implications?
Sure. So this antibody arming system we call ACC stands for antibody cell conjugation. So antibody is a tumor targeting molecules. So it can recognize and then bind to tumor cells. And then for what we do is that we use a simple conjugation method to conjugate those antibodies to the immune cells, such as NK cells. In that the NK cells is one of the immune cells in our body. It's an army of our body. However, intrinsically, they are powerful, but they don't really have the targeting ability within them. So the ACC allows us to conjugating those targeting device to the NK cells, and then for them after that, for them to be able to target tumor cells. That means that they can now recognize tumor cells, bind to the tumor cells, and then kill the tumor cells. So those targeting device we call antibody, we can conjugate to the NK cells. We're not conjugating one or two, we're conjugating hundred thousands. So we're able to put hundred thousands of those targeting devices on the NK cells, so that so they can recognize target tumor cells and then kill the tumor cells. Try to restore and then enhance the ability of the immune cells for them to fight and beat cancers.
Now, could you expand on Acepoida? We know that you've developed a proprietary platform. Could you expand on that?
Sure. So leaning towards that, we using the ACC platform, we were able to conjugate hundred thousands of antibody to the immune cells. This is very important in that it overcome one of the big challenge in cancer biology. For that cancer cells, they express we call tumor antigen. So they express those tumor antigen. They have the high expression and then low expression. And then most of our current drug, they can kill the high expression tumor antigen cells, but not the low expression ones. However, for what we do, we conjugate hundred thousands of the antibody on the NK cell, and then these hundred thousands of antibody can easily grab the tumor antigen expressed on the tumor, even they express in a very low number. So that allows us to now can target and then kill both high expression tumor antigen tumors and then also low expression tumor antigen tumors. So that in that way, we will have the potential to wipe out the whole tumor population in that they will not be able to relapse or remit. So that will be the kind of we see the one of the big strengths of our technology in that we try to address right now a big challenge in oncology.
Now, the ACC platform, it's potentially more cost effectively link antibodies to immune cells, hence augmenting or enhancing their tumor killing ability without the need for genetic manipulation. Can you share why that is important with our audience?
Yes, so our ACC again is using a very simple chemical conjugation to link the antibody to the NK cells, hundred thousands of them. So first, this it can only be done through our chemical conjugation. The genetic engineering usually allows 1,000 to 10,000, but not able to up to hundred thousands. So that first is that we can put really more targeting moiety on the targeting device on the immune cells, so that recognize low expression tumor antigen tumor cells. And then second is that the genetic engineering, you do it once, you change the genome once. So every time you do it, every gene you insert into the cells increase the risk of the unwanted genetic information. So that the genetic engineering it's powerful, but it also comes with the risk in that it is changing the genome of the cells, and then more times or more gene you put in, the higher the risk you bring in the unwanted genetic information into the cells. So that the aspect to us is that our ACC, since it's only conjugating on the cell surface membrane protein, we get the target device on the membrane and didn't change the genome of the cells, so that we also have less risk and more safe.
which is always a good thing. Now, I want to talk to you about your ACE NK. Can you tell our audience a bit about that?
Sure. So ACE-NK stands for the antibody cell conjugation NK cells. So that means we conjugating anti-tumor antibody towards the NK cells we produce. And then those NK cells are produced in that they are a stable NK cell line with high expression of the activation receptors and minimal expression of the inhibition receptors. So that's another advantages or another big figures we have to overcome the challenges in current cancer biology. In that the other reason why the immune cells doesn't work in cancer patient in that the tumor cells can inhibit immune cells. However, through our ACC and then our proprietary NK cells, so NK cell, we make them for them to have high expression of activation receptors. And then we link, using the ACC to link those antibody to the activation receptors of those NK cells. The result is that when we get our ACE NK cells made, and then for them to engage the tumor cells, they are greatly activated and then can override the inhibition signal from the tumor. So those are really two of the biggest challenges we try to address. One is we can target low expression tumor antigen tumor cells. And then the second is that ACC can guide the NK cells towards tumor cells. And then when they engage the tumor cells, greatly activate the NK cells. And then that activation can override the inhibition signal from the tumor cells, and then can have the NK cell kill the tumor cells. So those are the challenges we try to address, and then using our technology platform, hope for eventually bring a more potential therapy for the cancer patients.
Now, shifting over to talk about your ACE 1702, the anti HER2 conjugated NK cells, they have been validated in vitro and in vivo, presenting promising treatment possibilities in a variety of cancer types. Can you expand on that?
Sure. So our product is 1702. It's conjugating an anti-HER2 antibody to the NK cells to target and then kill HER2 expressing tumor cells. And then this is very exciting and then also encouraging in that this 1702 we made, they can target and kill high expression, HER2 high expression tumor, and then also HER2 low expression tumor. So with the HER2 expression tumors, they actually occur in many different cancer patients: lung cancer, breast cancer, endometrial, ovarian, biliary tract, gastric, colorectal, salivary gland. So many of them. So it's a very important tumor marker. So the tumor cell express those markers. However, the expression level is different. There's a high expression we call 3+. There's a medium expression we call 2+. And then there's low expression we call 1+. And then our current treatment can only target 3+ or a little bit 2+. However, left out the 1+ and then 2+ tumor cells. So that's where our 1702 can come in that, as we mentioned, we can conjugate more than 100,000 of anti-HER2 antibody to the NK cells. So that even is 1+ HER2 expressing tumor cell, low expression, they will still be recognized and target because we have 100,000 of those targeting devices. So even they express a little, they will still be target and then recognized by the 1702. So that's what we hope to address these challenges in this we call dynamic expression of the HER2 expressing tumor, so that in the end, hopefully we can kill both high expression and low expression HER2 expressing tumor cells.
Which would be fantastic. So let's talk about what are the biggest challenges that you think you'll be facing in the drug development effort? If all goes according to plan, when could we expect to see a new biologic application filed with the FDA?
Sure. So the biggest challenge we have seen from our experience will be still well, mostly on the we call CMC, so the chemistry manufacture and control. Same for us, and then for a lot of the cell therapy company in that making a cell therapy drug in an R&D level is easy. However, make them to be the scale that can be used to actually treat patient, that's much more difficult. So that it also take us more than a year to iron out our CMC process, the production process, in that we can make the GMP clinical grade cells as a drug to treat patient. So that early this year, January this year, we got a clearance from US FDA, so that they see that we were able to produce quality and potential cell therapy drug 1702, and then now we're running the phase 1 clinical trial in the US, including the medical center such as MD Anderson. So the CMC is challenge. However, I think luckily for us, with our team effort and our senior management have tremendous experience in manufacturing the cell therapy drug, so that we can overcome these challenges. And then currently we're running the phase 1 clinical trial in the US, and then we expect we start our phase 2 trials next year, 2021, and then the potential BLA can be as early as in 2023.
Now, what about the company's financing? Have you raised all the capital needed to develop the drug candidates, or are you looking for more funding?
So right now we are not actively raising funding. We have been raise funding, and then I would say it will be sufficient for carrying our products forward. And then also not only the investment, we have also have a partnership deal, so that we have also we call non-diluting funding going in through the partnership. And then so with those combined, we see that we will have sufficient resources to push our product, not only the 1702, but the second one as well, into the clinics.
And then could you talk a little bit about the clinical trial pipeline on that note?
Sure. So as we kind of mentioned previously, 1702, the anti-HER2 targeting NK cells, as our first pipeline, and then currently in the phase 1 clinical trial in the US. And then our second pipeline potentially will go into the trial in the US as well next year, 2021. Currently, we have a couple of products that's showing very good preclinical efficacy. One is the CD70, the other one is PDL1. So CD70 for we're using the same platform, so we're conjugating the anti-CD70 antibody to the immune cells, and then that product can target both liquid and solid tumors. So for example, renal cell carcinoma, ovarian cancer, those cancer type have the CD70 expression, but also the AML and lymphoma, they have the CD70 expression as well. So this is also, as we see, very potential product that can be used in variety of different indications. PDL1 as well. So in that concept, we were conjugating anti-PDL1 antibody to the immune cells. And then PDL1 also can target variety of different tumors, including lung, including liver, including melanoma, and then many others. So those two pipeline, we expected they will go into the clinical trial next year or early 2022.
Now, before we let you go today, any final thoughts for the Trial Site News audience?
So I think we just start to see the emerging therapies and then treatment that can further extend patients' life and health. And then for what we do, really, we see that the immune cells be our own army to fight cancer since we were born, and then they have doing that job pretty well until we get old. So the concept for us and then for many other immunotherapy company is that we try to enhance or restore the function of our immune cells, and then we are the pioneer at doing this enhancement at the interface of chemistry and cell biology. So we hope with this novel technology we can bring in more enhanced and then amplified immune cells to help the cancer patient to fight and beat cancers.
Well, Doctor, thank you so much for your time. We know you're busy and we appreciate you taking the time out of your schedule to come talk with us.
Yeah, thank you very much. Thank you for your time as well.